So today we're going to cover two things. The first is DNA structure. I went over that, and you probably don't even remember because it's like online learning, blah, blah, blah. But I did cover the structure of nucleic acids in chapter 3. This is probably the first or second unit that we studied in grade 10. Anyway, we looked at the three main experiments that led to the conclusion that DNA is in fact the hereditary material. Just to review, we had the Griffith experiment where he discovered transformation in bacteria when he heat killed the pneumonia-causing bacteria and he mixed it with the live benign or non-disease causing bacteria. Something transferred from the heat-killed bacteria to the benign living bacteria and turned it into the virulent strain. That was called transformation. Then you had the Avery experiment where he used enzymes, 3 different enzymes that break down DNA, RNA, and protein respectively, and mixed he mixed bacteria from Griffith's experiment and found that transformation did not occur when the DNA destroying enzyme was used. And then finally, we have the Hershey-Chase experiment with the bacteriophage. Hold on a second, let me let Pete in. There we go. Is that everybody? Nope, still one more. That's okay. And then finally, Hershey-Chase with the bacteriophages when they labeled the protein and the DNA separately. And then when they did the centrifuge to find out which material was inside the bacteria, being the hereditary material, they found out that it was DNA. Oh, okay, and there's bang. Now we're all here. Yay. So the first topic today is the structure of DNA. So by the early 1950s, most biologists had accepted that DNA is the hereditary material. However, they still didn't know what the structure of DNA was or how it could replicate, store, and transmit the information and direct cell function. So they know through experimentation that DNA is the hereditary material. We just don't know how it works. These mysteries would soon begin to unravel at Cambridge University in England. Now, these are probably in biology the 2 most well-known names. These are Watson and Crick. This is in the 1950s at Cambridge University in the UK. They teamed up to determine the structure of DNA. It turns out that my professor in university that taught me molecular genetics actually knew these 2 guys or worked with them or something like that. I can't remember exactly. He died a couple of years ago, I think, but yeah, so that was cool. By 1953, they proposed, that DNA is made of 2 chains that wrap around each other in a shape called a double helix. That is a winding staircase. Now, a big part of their discovery involved Rosalind Franklin, because they obviously didn't do it all by themselves. I think they were biochemists, so they helped to determine the bonding inside DNA, which determines its shape. But here we have Rosalind Franklin, and she did X-ray diffraction photos of DNA crystals. That was Rosalind Franklin and Maurice Wilkins that actually took, this is an actual, one of the first photographs of the DNA molecule, and you can see how it crisscrosses there. This is what helped Watson and Crick determine that it was not only 2 chains that were connected in the middle, but that it was actually twisted, giving it that double helix shape. In 1962, like 9 years after this discovery, Watson, Crick, and Wilkins received a Nobel Prize. And I know you might be thinking, oh, well, of course, it's the woman that doesn't get the recognition that she deserves. It's not actually true. The reason why Rosalind Franklin didn't get it is because she had died 4 years earlier, and they don't give Nobel Prizes to dead people. So it's called posthumously. They don't give it after somebody has died. So she is given recognition for her work through teaching and things like that, but there's no actual prizes given. But it was basically those four scientists. Now, when I taught you about DNA and RNA as a nucleic acid, remember these are nucleic acids. So DNA is deoxyribonucleic acid, and the deoxyribo comes from the name of the sugar. When I taught you that, it was in chapter 3 when I was talking about biological molecules like carbohydrates, proteins, lipids, nucleic acids. And the key to biological molecules is that they are made of repeating subunits. And each of those categories of compounds, carbs, proteins, lipids, nucleic acids, they have a base unit that repeats itself over and over. For DNA and RNA, the base unit is called a nucleotide. And you can see the 3 parts of a nucleotide in this diagram. We have the sugar molecule and the phosphate group. Now this makes up the sides. If you think of DNA as like a winding ladder, that's a terrible double helix, but anyway. And then you've got the steps in between. So if you were to untwist it, it would basically look like a ladder, right? This is what makes up the sides of the ladder. So it's alternating sugar, phosphate, sugar, phosphate, sugar, phosphate. And then the quote unquote steps of the ladder are these nitrogen containing bases. Now this would be 1/2 of the ladder, and then you'd have a hydrogen bond here, and you'd have another base on the other side. And then you'd have, oops, go on. You'd have another base on the other side, and then attached to that, you would have your sugar. So that's terrible anyway. And then your phosphate. That's the sugar phosphate. So you got the phosphate group here, sugar, phosphate, sugar, like that. So on both sides of the ladder is alternating sugar and phosphate groups. Attached to the sugar is the nitrogen containing base. Now, the reason why it's called DNA is because the sugar in DNA is called deoxyribose, like I said. Okay, so Watson and Crick were biochemists. They were able to determine the double helix shape because of their knowledge of bonding. So the alternating sugar phosphates are the sides of the handrails of the staircase. I guess they use staircase, not a ladder or whatever. The nucleotides are connected by covalent bonds between the sugar and the phosphate. So you can see from the diagram that the bonds holding the sides of the staircase together are covalent bonds, but the bonds holding the steps of the staircase are hydrogen bonds. This is really, 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 really important. AKA, if you have your book or whatever, you should be highlighting that, because there is a very important reason that the sides of your ladder are, let's just call it basically unbreakable, unless you're talking about temperatures of around 45 degrees or higher. And then these weaker bonds that you have holding the sides together, it has everything to do with how DNA copies itself. Okay, I'm going to talk about that a bit later, but really, really, really important that you understand that the sides of the stairs are held together with covalent bonds, but the steps are hydrogen bonds. Hydrogen bonds are weaker. They're easily broken. Keep that in mind for when I talk about DNA replication later. You do not have to remember how many pairs are in each turn. I just gave you that as a reference if you want to understand the length. A really good analogy for understanding how long DNA is that if you were to expand the nucleus, just the nucleus of a cell, to the size of a basketball, DNA would be 64 kilometers long. Okay, let me repeat that. If the nucleus of your cell was the size of a basketball, the length of DNA would be 64 kilometers, not meters, kilometers. It is an extraordinarily long molecule compared to the size of the nucleus. And keep in mind that every single one of your 30 trillion cells in your body has a complete copy of DNA. Now you might think, and this is the key, this is the whole point of the genetic code. And like the next couple chapters, we're going to talk about how the information on DNA is translated into like physical things. Like how does it create different kinds of cells? How does it create like our eye color? All of that kind of thing. The information on DNA is a code to make proteins, and proteins are basically responsible for everything in biology. And you have over 200 different types of cells in your body, and each of them have different functions. So let's say a liver cell is going to access and activate certain parts of DNA, whereas a nerve cell is going to activate completely different parts of the DNA. Having said all of that, our DNA sequence, because of the Human Genome Project, which occurred in the early 2000s, it took about 20 years. Many international organizations working together to map out the exact sequence of nucleotides in the human genome and other species as well. It turns out that our DNA molecule, the sequence of bases, is 98% identical to our closest relative, which is the chimpanzee. 98% identical. That means the only thing genetically separating humans from chimps is 2% of our DNA. That's it. It's crazy when you think about it. And when they were doing the Human Genome Project, they actually discovered that 80% of our DNA is not used. So imagine the instructions that are in our DNA, like that connects us to our ancestors all the way back to like Australopithecus. Like if you've seen the movie Lucy, when you've got like a 1 meter tall humanoid, I say humanoid because it's not a human, because there's not the genus Homo. The genus was Australopithecus. It's our ancestor before humans, covered in hair, walking with their shoulders stooped over because we weren't completely upright yet. That only occurred when Homo evolved. But we share so much DNA. I'm sure there's probably still genes in our DNA that if activated, we would also be covered in hair. And there's actually a genetic anomaly where that occurs. And I can't remember what they call it, like Wolf syndrome or something, where like their bodies are just completely covered. It's fascinating how much we don't understand about our own genetic code. Anyway, we'll get to all of that good stuff in the next couple of chapters. Okay, so here we have a DNA molecule, which is still in its helical form. So you can see here's the base attached to this strand. We have C, A, T, and there's G. There's 4 bases. You see adenine, guanine, cytosine, and thymine. These 2 are where the hydrogen bond keeps the stairs of the DNA helix together. So these form the stairs or the steps, hydrogen bonds. All right, now the bases, as I said, adenine, guanine, thymine, cytosine, they come in 2 pairs. Now, adenine and guanine are double rings. You can see there's double rings here. These are called the purines. And the single rings are called pyrimidines. Now, the way that I remember it is gap, guanine and adenine are purines, although they both start with P, so gap pure, I don't know, whatever. Any way that you can try to remember that. We could do pure ag. I don't know. Just to try to think of a way that you can remember which ones are purines and which ones are pyrimidines. Picked, maybe? Piked? I don't know. Pyrimidines or cytosine and thymine. You can figure out a way. That's a really easy way to remember groups of things, is trying to come up with an acronym to do that. But anyway, the pyrimidines are single rings. The purines are double rings. Those are the 4 bases. Now, there's 2 rules when it comes to base pairing behavior. If you go back to this one, you might notice that every time you see a cytosine, it is paired with a guanine. So if you see here C and G, C and G, C and G, G and C, You see a little bit there. And A and T. AT, 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 AT. These are what are called the base pairing rules. These two pairs always pair together. So C always pairs with G, A always pairs with T. This is how DNA replication is going to work according to the base pairing rules. And I'll talk about that again a little bit later. So these two are called complementary base pairs. They complement each other. So A complements T, C complements G. So this is another example of the base pairing rules. So if I were to white out this entire strand, you could use the base pairing rules to complete this copy of DNA, because when there's A, the complementary pair is T. When there's C, it's G. G, when it's T, A. G is C, T, A, G, C, A, T, G, C, A, T, C, G. Okay, always A, T, C, G. This is what's called the base sequence, the order of bases on a chain of DNA. This is part of what's called the genetic code. The sequence of bases is going to determine the sequence of amino acids in a protein chain. If you remember all the way back to chapter 3, proteins, the base unit of a protein, because it's a macromolecule, is an amino acid. And amino acids are made from nucleotide bases. So the sequence of bases is going to determine the sequence of amino acids, which is going to determine the type of protein you make. That relationship is called the genetic code. If you substitute one of these, let's say instead of a C, you put a G there, that means that this becomes, oops, C is green. Now you've completely changed the order of amino acids because you've changed one nucleotide base. It's that easy. Okay, so now we get into replication. Now, Watson and Crick, along with Wilkins and Franklin, their discovery of the double helix created a lot of excitement, obviously, because once you know the structure of DNA, you can start to look at it and how it relates to the process of heredity. So using the model of the double helix, you can explain very easily how DNA can replicate exactly each time a cell divides, which is a key feature of hereditary material. You can't pass on your genetic information if you can't copy DNA. And it's really, really simple, okay? Remember I told you, it's really, 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 really important for you to understand why the bonds holding the bases together are hydrogen bonds that are easily broken. This is why. You're basically, if I just to sum up DNA replication in a couple of sentences, you're going to use an enzyme to unzip or to break the hydrogen bonds in the middle. And then you've got another enzyme that's going to come along and read the template strand, use the base pairing rules to add the complementary base to the other strand. It's going to happen on both strands separately, going in opposite directions, which is what I'll talk about in a minute. And then you're going to end up with two completely new strands. Okay, so if you follow that, fantastic. If not, don't worry. I'm going to go through it step by step. Okay, so replication is the process of copying DNA before the cell divides. If you go back to the cell cycle, and I know you're like, oh my God, we've got G1, S, and G2, then we have mitosis, and then we have cytokinesis. This is where DNA is copied. Okay, G1 is the first growth phase that occurs right after cell division. So the cell just finished dividing. You've got two brand new tiny daughter cells that have to grow to a mature size. During the S phase, the DNA is copied. That's where DNA replication takes place. G2 is when you prepare the cell for division. Then you have mitosis, which is the division of the nucleus, cytokinesis, the division of the cytoplasm. So I'm going to describe how DNA is replicated in the S phase. So as I said, the most basic version is that the two nucleotide strands of the original DNA separate. Using the base pairing rules, you're going to use each separate strand as a template. You're going to read the sequence of bases on each strand, and you're going to apply the complementary base and bond that together, and you end up with two new strands. Because of the complementary base pairing rules, each strand serves as a template for a new complementary strand. Therefore, you end up with two complete sets of DNA. This method of replication is called semi-conservative replication. And the two scientists that proved this process are Messelson and Stahl in 1953. So it was the same year that Watson and Crick came out with their double helix model. That's how fast it happened. So what does semi-conservative mean? Well, semi means half. Conserve means to save. Now, if I were to draw the old strand in red, and the new strand in blue, you can see that the new DNA molecules down here are half red and half blue. So we have saved half of the original strand. That's semi-conservative replication.
The Messelson and Stahl experiment provides evidence for semi-conservative replication of the DNA molecule, where the two parent strands serve as the template for synthesis of new strands. In this experiment, bacterial cells were grown for several generations on a medium containing a heavy isotope of nitrogen, N15. The DNA in these cells therefore contained heavy N15 nitrogen. The cells were then transferred to a new medium containing the normal lighter isotope, N14. At various times after the transfer, samples of the bacteria were collected. The DNA was then extracted and dissolved in a solution of cesium chloride. The samples were then spun rapidly in a centrifuge. When the cesium chloride is centrifuged at high speed, a concentration gradient is established in the tube. DNA molecules move in the gradient until they reach a place where their density equals that of the cesium. DNA containing N14 moved to a position in the gradient determined by its density. DNA containing N15 is denser than that containing N14.
Okay, so what he's saying in the centrifuge, which you saw the tube spinning around fast, that means that the heavier items are going to be pulled to the bottom where the lighter elements will stay at the top. So you've got N14 at the top, N15 at the bottom.
So it sank to a lower position in the cesium gradient. After one generation in N14 medium, the bacteria yielded a single band of DNA with a density between that of N14 DNA and N15 DNA, indicating that only one strand of each duplex contained N15. After two generations in N14 medium, two bands were obtained, one of intermediate density, in which one of the strands contained N15, and one of low density, in which neither strand contained N15. Messelson and Stahl concluded that replication of the DNA duplex involves building new molecules by separating parent strands and then adding new nucleotides to form the complementary strand on each of these templates.
So what that means basically is like in the agar, the medium with the petri dish that had N15, You have bacterium because nitrogen obviously is needed for the bases, so you can follow the path of it that way. Both strands of the DNA contain N15. When you take this bacteria and you transfer it to N14, then that means that after one generation, the DNA is going to be half N15, half N14. And after the second generation, it's going to be 25%, because what I mean, the strands are going to be 25% N14 and 25% N15, 75% N14. So every generation after that, you're going to see less and less N15. And that's how they confirmed semi-conservative replication. Okay, so what are the steps of DNA replication? The first thing that happens is an enzyme called DNA helicase. Helicase separates the DNA strands. It's going to move along the DNA, breaking the hydrogen bonds between the nitrogenous bases. And I always use a zipper as a really good analogy for this. If you imagine the actual zipper part, when you unzip your jacket or your purse or whatever, your backpack, the point where the zipper is called the replication fork. Now, the word fork in English just means to split, right? If you talk about a fork in the road, you have the road that goes off in one direction and another direction. You've got two directions to choose from. So the helicase creates a replication fork. Now that means that you've separated the two strands, and that means that you have second enzymes that come in called DNA polymerase that are going to read each strand, and they're going to read the bases on the template, and they're going to add the complementary strands. The tricky part comes in where you talk about the direction that polymerase goes in. And I'm really not happy that we are online because it is so much easier to explain this in person. I have videos, but it just doesn't have the same effect. I might make a short video later with string. I usually use string to explain it. And I'll upload that to GC or something so you can watch that. So there's a bunch of nucleotides that are floating around inside the nucleus, and DNA polymerase is going to add complementary nucleotides to both strands from those free floating nucleotides and then form covalent bonds in the new strands here. So each new DNA helix, for each new DNA helix, one strand is from the original molecule and one of them is new, and that is semi-conservative replication. Okay, here's where mortality kicks in. Every time DNA copies, some nucleotides at the end of your chromosomes are lost. Some eukaryotic cells have nonsense sequences at the ends that repeat thousands of times. These are called telomeres. The telomeres are basically what cause aging. What that means is there's a certain number of times that your cells can replicate and that DNA can replicate before things just start breaking down. It's kind of like when you take a photocopy and then you photocopy the photocopy and you photocopy the photocopy of the photocopy, and you keep doing that. The copy each time becomes more and more degraded. That is what causes aging in all life on Earth, is the formation of telomeres. Thought I had a video on telomeres actually. Let me see. I'll have to go back and download that again because I used to have a really good video explaining telomeres. Anyway, all right, so here's where it gets complicated. When you divide the DNA double helix and you have the two strands, the way the bases are added on each strand, they happen in different directions. So on one strand called the leading strand, see if I can kind of draw this. So, oh no, like that. Okay. So here is your helicase at your replication fork. Now, like I said, you're going to be adding complementary base pairs, but it's going to happen in two different directions. So if DNA helicase is moving in this direction, there's one strand where you're going to add complementary bases in the same direction that the helicase is moving. This is called the leading strand, because you're just kind of adding as it gets separated, right? The other strand adds it in the opposite direction. That's called the lagging strand. Now, because, and this is the part that's hard to explain without showing you, because you're adding the bases in the opposite strand, you basically have to wait until, let's say you've already added these segments and helicase is here. You have to wait for helicase to unravel a new section before you can come back up here to add these going in this direction. I don't know if that makes sense, but like I said, I have a video. I will, I don't have any string up here right now, but I'll see if I can help clarify that later. Okay, so as the fork moves along the original DNA, the synthesis of one strand follows the movement of the replication fork. That is the leading strand. The other strand is going to move away from the fork. So again, you like imagine if you are unzipping, I don't even have anything with a zipper. Do I? I don't think so. Imagine you're unzipping your hoodie and you're going to add a zipper, but going up. You have to wait until you unzip to add the new zipper, if that makes sense. So this is what I was talking about. So if the direction of helicase is going to the right, the top strand here is the leading strand because it's going in the same direction. So as you unzip, you just keep adding more bases. But the bottom strand is the lagging strand. It's going from the 3' to the 5' direction. You can see here it's labeled 3' to 5'. And this one is also 3' to 5', but they're labeled in the opposite direction. So if you only unzip up to here, then you can add this section, but you have to wait for helicase to get up here before you can add this section. So you end up adding the bases to the lagging strand in chunks called Okazaki fragments. So you have chunks of nucleic acids, or sorry, nucleotide bases that are in the nucleus. Let's say you've got like an AAT chunk and you've got like a CG chunk or a TC chunk. You can add these chunks that are complementary to the strand, and any of spaces that are in between these chunks, you use something called an RNA primer to kind of hold the place while you're adding the complementary bases. So the RNA primers are going to fill the gap, but eventually they're going to have to be replaced, and those are joined later by an enzyme called DNA ligase. I'm sure that's all quite confusing, but this is going to be a computer animation of what DNA replication actually looks like. So if you were able to zoom in, if it's going to work. No, that didn't work. Oh, hey now. Here we go.
Using computer animation based on the latest research, we are now able to...
Okay, so this is based on research. This is your double helix. It's being fed in, use the laser. The DNA is being fed in, this is helicase right here. And these are the polymerases here. So this one is the leading strand. So you can see that it's adding bases in this direction. because it's coming out as a continuous stream of a new double helix. This is the lagging strand over here. So you're going to see one of these enzymes, I think it's the green one, coming over here and pulling out a big string of separated DNA and then adding it going backwards.
How DNA is actually copied in living cells.
You can see it going
You
the
are
other
looking
way.
at an assembly line of amazing
See how it's going that
miniature
way?
biochemical
And then it adds it up
machines
to the
that are
previous
pulling
section.
apart the DNA double helix.
This one is the leading
and cranking
strand.
out a copy of each
It's just adding it
strand.
as it comes out.
The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called Helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously
That's this
and
one.
can be seen spooling off to the right.
The leading strand.
Things are not so simple for the other strand because it must be copied backwards. It is
You can see it
drawn
going
out
backwards.
repeatedly in loops and copied one section at a time.
You see it now copying a new section,
The
going
end
back
result is 2
to meet the
new
previous
DNA molecules.
copied section.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called Helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA.
Okay, so the reason why the primer is put there is as a signal for polymerase as to where to start. Because remember when I told you that if DNA is the size, if the nucleus is the size of a basketball, then DNA is 64 kilometers long. Well, we don't need all of that information translated all of the time. We only need a tiny, tiny little section. So how does polymerase know where to start unless there's an indicator there? So the primer acts as an indicator, like, yo, start here.
DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments.
I'm assuming that there's some Japanese scientist named Okazaki that was the one that discovered the lagging strand chunk adding part.
Each fragment is started with an RNA primer.
Same as the leading strand. But you can see, because the lagging strand, you've got the primer from the previous section, followed by the copied, or not the copy, well, yeah, I guess the copied strand. But now you've got a new section that came out, and now you've got to put the primer for like, oh, here's where we start the new section, and then add the bases here. No. Oh, crap. hold on, I'll skip forward.
Bases one by one in the
Yay.
chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA.
These are the gaps I was
Another
talking about.
DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay. Now, obviously, there's a difference between DNA replication in, oh, I got 2 minutes. Okay, prokaryotic and eukaryotic replication, because a prokaryote DNA molecule is smaller and circular. So basically, it starts in one place and moves to the other end. So if you have a circular molecule, your helicase will start here and move in opposite directions until they meet on the other side, giving you two new loops, like two zippers going in opposite directions. This will show you how that works. So the red and the blue are the two strands of the double helix. Okay, replication, the helicase is going to start here. I'm going to separate the two strands, and the dotted line represents the complementary new strand. And boom, there you go. You have two new DNA molecules. Eukaryotic DNA is obviously really, really long. For the human, we have 46 chromosomes. There's even some organisms that have over 1,000 chromosomes. If DNA replicated the same way that, if DNA in eukaryotes replicated at the same rate that prokaryotic DNA would, it would take 53 days just to copy the biggest chromosome, let alone the other 45. So obviously we cannot copy DNA the same way that prokaryotes do. We actually have thousands of replication forks. So you can imagine there's thousands of helicases that are separating the DNA at thousands of different locations to make it quicker. And what happens if there's an error? Generally, there's not errors. Out of every 10,000 paired nucleotides, there might be one. But DNA polymerases also have the ability to proofread to make sure that there were not any mistakes. If a mistake happens, that means that the base sequence of the new DNA doesn't match the old DNA. That is called a mutation. Sometimes errors don't get repaired, and that's when you can have a bigger risk of cancer and things like that. And I got to finish because class is over. I will